So I have a couple different questions that I've started writing down, and then maybe I'll ask Brent to help me find questions in the Zoom Q&A. But first, I want to start out with like a general question for all three of you, and maybe I'll start with you, Sanjay, because you kind of alluded to this at the end of your talk, which is that in especially it seems like the spatial transcriptomics field, there are all of these technologies that are coming out at like rapid fire pace, but they haven't really, to use your word, like penetrated yet, right? Like there's still only a few labs that can use these techniques really effectively. And I'm wondering if you have any like thoughts or suggestions for people that might want to start using these techniques, but like don't know how to start. Like, is their best bet to just try and collaborate because it's, such an ordeal to get it started? Or do you see, SkySeq being employed by like multiple labs across the world?
Yeah, this is a great question. And as a tech developer, like I think we have a very different view of the world and the skill sets and the set of things we engage in. I think that really it comes down to moving these kinds of technologies from the academic setting into a product setting. So for sequencing, for example, as opposed to having to kind of pour sequencing gels and kind of do that next-gen sequencing in-house, like the founding Celexa and Illumina and so on, those companies were really able to democratize that access to that kind of component. And for a number of those technologies, there is kind of a commercial kind of product linked with that technology itself. So for example, I mentioned with Merfish, there's VisGen. For some of these kind of spatial spots, I think 10x has the Visium and the Visium HD coming out soon. I expect BGI to kind of commercialize their StereoSeq platform. So as these kind of different commercial groups kind of start to rival each other, I think one will kind of take over and hopefully the costs of that will drop. But I think that's kind of the best way in the long run. In the short run, definitely collaborating is very helpful if you know the people or if you can kind of manage that.
Yeah, thanks. I think that's a very interesting point about getting it to the product space. I think I've like seen that definitely in the single cell RNA sequencing, just not spatial, but like now we can get the kits that do the split pool split technique originally developed in the Shinduri lab, and it seems like that's really helping to democratize that technology. So, Lacra, do you have any?
Wait, just with respect to that question, I think always the microscopy is harder to adopt. Not only, just the image analysis is not at the point where the sequence analysis is. So that's another thing when it goes commercial, hopefully like the standardization, even data formats are not as standardized with images. And then the analysis of those spots and everything is just harder. So, in a sense here, what Sanjay showed is better because you rely more on sequencing after you barcode it. But still the initial, all of the sort of omics by imaging are just harder. It's not as standardized, even though even sequencing it's at its core imaging and detecting spots, but it's harder in cells.
Yeah, I think it would be great if we could move to like similar to like a PDB where you have like coordinates for each atom, or and I think I totally agree with you. Like the count matrix for single cell data has like really made it so you can integrate and kind of have a standardized format between different between different technologies. It's a really good point.
Yeah, maybe this is like kind of related to that question, but and we'll start with Gong here. Gong, did you have a point you wanted to make about the question?
It's indirectly related because in terms of the penetration point of view, third generation sequencing somehow actually make it easier because people no longer need to do a lot of biochemistry, like optimize different antibodies, enzymes. They can directly cause some of the modifications. So in that sense, it's actually making things easier. But sometimes easier doesn't mean always better because like some of the technology may not be ready yet for all kinds of applications. So people still need cautions when using them. So just a quick comment for that.
Well, that was actually going to kind of be my next question. So I was really struck by your talk. Actually, it's the second time I've heard you give that talk, because I heard you give it at the long read sequencing workshop a few weeks ago.
Cool.
But about this idea that, you know, the 6MA is kind of irreproducible. And can you speak to like the types of things that people should be considering when they're using these technologies, that, you know, like the types of controls they should use in both the experimental setup and in the analytical pipeline, specifically for third generation sequencing, but then I'd love to hear from Lacra and Sanjay as well.
That's a great question. So, and I mean, the full answer will be taking too long, but I will try to give some high level view of that, because at the high level, yes, it's exciting that third generation sequencing allow people to directly map these modifications. I mean, both on DNA and RNA, like in Sarah's talk in the first session. But I think people, some people at least, didn't appreciate the fact that if you can detect everything, you're not detecting anything. Does it make sense? So basically, if we can call everything, because it's not specifically detecting a specific thing, it's just detecting something is there. Actually, that's the right way to view third generation sequencing in terms of map these epigenetic marks, is that it's only telling you that something is there. So what it is, you need actually a lot of work out there to do. Some cases are obvious, because if you detect a lot of modifications in the human genome, it has to be 5MC, because no other modifications that abundant. And we study bacterial a lot, like say 6MA and motif driven. If you detect it in bacterial, yes, it has to be 6MA, 4MC, or 5MC. So, but as soon as you get out of these things, either abundance has some unique properties, or their distribution have unique properties, then like Mark mentioned, it become real like Wild West, basically. And at that time, it's basically people need to do a lot of extra things to really be sure that they're studying the things that they're detecting. So think about it this way. I mean, I will not take too long, but just the one last thing is, some of the papers published earlier, they are not just applying these technologies, say we find some marks out there. Actually, there's methyltransferase, demethylase, readers, specific disease conditions, specific cell types. So basically, I would want to put forward a view is which, if we without super caution, it's actually very easy to find something, but it's much harder to be sure that something is really going on. I think I will end there. But great question. Yeah.
Can I say something about the third generation technologies? Like we
I saw
also tried them. We love them. I think the kind of a complementary point to what you said in terms of just what modifications you detect. For us, since we perturb specific genes, we would really like to be able to not sequence the entire genome, but like look at those specific genes. And the problem with a lot of the third generation technologies is that you get very few reads back. So you might be only find one piece of DNA or one piece of RNA once in your entire read. And so you can't do any gene-specific statistic. Like if I perturb a gene and put modifications there, it's very hard currently to do the selection method for just that RNA or that DNA. So for the human genome, which is huge, with this third generation sequencing technologies, you cover very little of it. At least that's how it was a few years ago when we tried it. I'm not sure, maybe it's moving fast towards covering more, without having to spend a ton of money. But with the next generation sequencing, Illumina type, for the same amount of money, you can get a lot more reads back. And there are ways to enrich that work better for a specific gene, right? So.
Great point, Lacra. I mean, just one thing to add is indeed the throughput is the one challenge, but actually you could find something helpful, which is they use also Sinai, but done in vitro. You can actually target certain regions that can enrich, and so if you add a certain region, either genetically or epigenetically, you can indeed enrich those regions. You will still need quite a bit of starting material, but and total yield is actually lower. But the good thing is you can actually get more reads. I won't say a lot more because it will depend on specific regions, like where we want to cut. but I would suggest you give that a try. I mean,
we did actually give it a try,
Oh,
just to
really?
say it is actually tricky, because indeed the amount, if you're looking at the DNA level, it's so little of it, right? Because you only get 1 or 2 pieces from each cell.
got you.
And you just have very little material to load on the nanopore, and that kills you, because then it's like very little material. Like somehow, the last time we tried it, it wasn't optimized enough that we could get it to work reliably, but we haven't tried it in a while. So maybe now it works more reliably. But the problem is also you have very little material, right? Yes. And so maybe you need to mix it with carrier, but then you like clog your pores with other pieces of DNA, right? Because if you just get femtomoles, right? Otherwise, you need to grow vats of cells, right? Like hundreds of millions or more of cells to get enough pieces of DNA, right? So it's kind of a problem that's not necessarily specific to the nanopore. If you don't want to amplify, obviously, if you want to look at modifications, if you can amplify, that's fine.
Very much true.
Lacra, we had a question, which you answered, I think, in the Q&A, but it's kind of related to like thinking about controls to use in CRISPR screens and stuff like that. The question about how do you make sure that you get one lentiviral integration per cell? Do you want to talk a little bit more about other things one should consider when doing screens?
So related to the lentivirus, usually it's just Poisson statistics. You infect with very little virus to make sure each cell either gets 0 or 1 on average, just based on statistics, and we kill the cells that don't get anything, because what we deliver has an antibiotic, in quotes, for an antibiotic resistance. So we kill the cells that don't receive anything. But again, for the screens that we do, like for measuring so many protein sequences, you also have to grow, which I didn't show, you have to grow a lot of cells, right? Because even though each cell is the test tube, you need to have about 100 cells that have each sequence. So that means if you do, but usually probably 1,000. So you end up having to grow hundreds of millions of cells. So we grow them in these big flasks, and that's kind of tricky, right? If growing and keeping them happy, and you can only do it with certain cell types that grow very well. So it is actually hard to do it in, like iPS cells or in an embryo. But people are moving now towards running this kind of experiments, even inside of an embryo, but it's trickier, I guess.
This whole, like, you need to have enough coverage in order to be able to do accurate statistics and infer anything, I think is really important, and is probably also really important in spatial transcriptomics. Sanjay, I'm curious, like the analysis pipeline for spatial transcriptomics seems hard. Am I right in that? Is there like, what are there things people should be looking out for when they're thinking about starting these types of analyses?
I think just like in the single cell world and other kind of sequencing-based technologies, there have just been so many groups that have put forth really nice packages and ways of analyzing this data. And it really comes down to like looking at which sets of genes are co-localized, or if you have imaging data, there's certain packages. But I guess like it always helps us start with a spatial hypothesis. I think spatial is the next trend of things where people may, say, what if this is a spatial phenomenon, and just kind of go forward that way. Having hypothesis first always just helps like narrow down the actual analysis you want to do. And luckily those are being standardized now by a number of groups. Yeah.
Cool. Is there anything I'm missing in the internet world, Brent, that I should be asking? I have more written down that I can ask, but.
No, you're doing great. And
Thank
there
you.
was one more question in the chat that I think can be addressed fairly briefly regarding what is the use of single cell technology right now in the cancer field? Well, that was a game ender.
I don't know of any, I know there's like a number of like clinical trials or preclinical things, but I don't know of any licensed kind of diagnostics that use single cell sequencing. It is very fast moving, but At the moment, I'm not confident I know of one.
Yeah, and single cell is certainly being used in the context of generating genomes out of cancer cells, and once again, starting to put together some of the architecture of the higher order cancers. But I didn't know if there was anything that perhaps any of the three of you wanted to add to that.
There has been work done, I think, to like just do standard single cell RNA sequencing of tumors. I think that's
absolutely,
you know,
tons of it, in fact.
Tons of that. And even cell lines, which actually was another kind of question I had that I was struck by when Lacra was talking, maybe not struck by, but you know, that even like tumors are obviously extremely heterogeneous. Developing organisms are extremely heterogeneous, but even like a cell line in a dish that you think is supposed to be kind of one thing is actually quite heterogeneous. And yeah, I don't know if I really had a question, but that was more just like interesting. And people are starting to do more and more single cell RNA sequencing, even of just standard cancer cell lines, and finding that these things are even more heterogeneous than we think. So there's like stochastic heterogeneity, cell cycle heterogeneity, and then like heterogeneity, other types of heterogeneity we don't even fully understand.
A question.
I mean, my understanding right now with the cancer diagnosis, that at least talking to genetic counselor friends, is that a lot of it is just mutations, right? Like they sequence the DNA. So for the cancers that are known to be associated with genetic mutations, they're standard. You can, I guess, PCR that region and see if it has the mutation. But for anything, even in cases where DNA methylation, for example, is known to be at a locus and specifically associated with particular types of cancers, it's very tough. And actually the health companies don't even approve those tests very easily because they're very expensive. So I feel like in many cases, even when the research has been done academically, it hasn't quite my understanding, at least from just talking to people who work in the field, it seemed like they don't often do any of these tests, anything beyond just sequencing the DNA. Just is not quite there yet.
I think that's true, Lacra. And so a question for you and Gong, I think, and Sanjay, feel free to jump in, but in terms of the epigenetics, and we just heard from Mark Akeson that we're now at a potentially 12-hour turnaround on whole genome sequencing for clinically relevant applicability. Where do we stand on that in terms of epigenetics, and what's, apart from maybe some HDAC inhibitors, what are we looking at going forward? And I realize this is some speculation.
I mean, in terms of diagnosis or in terms of treatment, because we're both.
Go for both.
I can start. I mean, I remember reading a paper saying that for a brain tumor, one of the important thing is to identify the specific type of brain tumor and subtype of brain tumor, so that they can tailor their therapies. So and the time matters. So I think that paper is get the tumor tissues, and that they can really have a much faster turnaround onto non-opera sequencing. You can really dedicate it to this purpose. And you can have these methylation patterns, and that they have a training data set, basically different type tumors, and you have a signature, and it's actually in terms of classification that's on the like easier end of the story. So that's one of the example. Of course, some people are using like liquid biopsy. That's another direction that people are going after. I think turn time definitely matters, and in that sense, non-opera sequencing has some very unique advantage.
And in fact, just within the last few weeks, the clinical diagnostics lab at JAX launched an assay using the Illumina EPIC methylation array combined with a classifier for CNS tumor types that was developed at the University of Heidelberg. And that's, I think, a leading edge of tumor classification, particularly in the brain tumor space. And it is true that the methylation characteristics do affect treatment strategies that are appropriate for those tumor types. Yeah.
And the array is much cheaper.
The array is much cheaper, yeah, and reproducible in terms of the content. Yeah. But I think that's likely to be extended into other tumor types as well. I think there's a lot of promise there.
Kaia, do you have one or two last questions? And then I think we're wrapping up.
Well, yeah, okay. My last question was just going to be to have each of you describe the what's your pie in the sky biological question that you want to address with each of the technologies that you're either working with or addressing. So I don't know who wants to start. Gong, you're unmuted.
Yeah. Okay. So I was starting. I thought I would go with the other. So, I mean, it's essentially the same as what Brent and Mark has said. So, because our expertise is really lies in the technology to map the epigenetic variations, and also on RNA there's a splicing. So, and we really want to build on these long reads and these modifications to better resolve regions that would have been challenging using short reads, and we want these to guide either screening or precision medicine. So that's our goal.
I think my pie in the sky would be kind of a in silico kind of movie or model of development, effectively, that's resolved at the molecular level, such that, if you were to put in some kind of mutation or knock out some cell type, you could have a reasonable guess as to what that would do, and start to do some kind of in silico biology with that.
I guess. And I would be happy if we can just really, when you put a new component into cells, you can predict what it does in a particular cell type and at a particular locus. Or you just put a new genetic circuit into human cells, and it actually behaves like you think it behaves. If you can build it and it works like you think it should work, then it means you really cracked it and understood it. Or not there yet. Even with even small synthetic circuits, right? Because I feel if we are going to go and do gene therapy, maybe not for cancer, but for diseases that are debilitating. And so just even being able to do that kind of control reliably, either with new pieces of DNA that we put in, that we can control how much it expresses, when, where, or just targeting genes in the genome, and we can actually control one gene and not affect the ones nearby in the 3D genome. And again, we have control of how much you turn it on and when, in what cell type. To me, that would mean we understand it. If we can build it or if we can control it, I know we understand the transcriptional control at least. So that's my desire.
Those were great answers. Thank you.
Kaia, thank you so much for moderating this session, and to each of the speakers. I'm going to turn it over to Brent for a couple of closing words and throw up the slide. He's been hiding in the background, but he's been the outreach coordinator for the Tech Dev Coordinating Center, and it's really his efforts and group and working with his committee that has made this session possible. So Brent, you want to close us out?
Happy to do that. So first and foremost, thank you so much to all of the speakers, as well as to Kaia and Mark for moderating. I thought this was a really good session, really informative, and I really appreciate, we all appreciate that you gave the explanations that I think lots of people who are just thinking about doing these sorts of techniques or are interested in these techniques could grasp and could hold on to. So hopefully this helps to move stuff forward. So thank you so much. Thank you to NHGRI for putting this on. And I think we are pretty much wrapping up today. So as part of the TDCC, we were happy to host this. And I hope everybody in your relative time zones has a good rest of the day.
You will get a survey. Let us know what we've done that you've liked and didn't like, and give us suggestions for future events. Thanks, everyone.